Dear students, until now we have studied the phase of the glycolysis which is energy demanding, that is in which we invest energy. तो आपने देखा कि अभी तक जो ग्लाइकोलिसिस हमने पढ़ी है उसमें हमने एनर्जी लगाई है in the form of ATP. 2 molecules of ATP have been utilized. Now let's move to the second phase or the next phase of glycolysis, which is energy yielding phase. The energy yielding phase starts with the oxidation of glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate, if you remember, was formed in the previous section, that was the energy demanding phase of the glycolysis. If you can see here that glyceraldehyde 3-phosphate after oxidation gives rise to the molecule 1,3-biphosphoglycerate. You can see here there is an additional phosphate group which is attached here now. This additional phosphate group will later be used when we will need to have a yield of ATP. In addition, another very important part of this reaction is that the NADH2 is formed here. So, this is why it is an oxidizing reaction, because NADH2 is formed here. So, that is why we also say that this is part of the aerobic phase of the glycolysis, because if this NADH2 is going to be used in the ETC, electron transport chain, it will require oxygen. The enzyme here is glyceraldehyde 3-phosphate dehydrogenase. And of course, this is the inorganic phosphate which we use here, which gives rise to the formation of 1,3-biphosphoglycerate. Then we have another very important reaction which have certain variation in different cells also. The 1,3-biphosphoglycerate then gives rise to the formation of 3-phosphoglycerate, a very important reaction, because in this reaction you get an ATP molecule. The ATP, the phosphate which was used or which was embedded into the last reaction in the 1,3-biphosphoglycerate molecule, it is now used for the formation of ATP. And the enzyme used here is 1,3-biphosphoglycerate kinase. Keep in mind that this kinase reaction replaces the true ATP molecules which has been used. So, 2 ATP molecules has been used in the previous section in the energy demanding phase of the glycolysis. The 2 molecules of ATP are not formed. So, it is balanced out. And you can also see here that there is another, if you can see this one, the formation of 2,3-biphosphoglycerate is another reaction. I will let you know about it very soon. Yes. So, 2,3-biphosphoglycerate, what does it mean? The 2,3-biphosphoglycerate is formed in the red blood cells, in the RBCs. You know, RBCs are specialized for carrying oxygen from one part of the body to the other part when the blood is transporting from one region to the other. And 2,3-biphosphoglycerate in this way increase the efficiency of the transportation of oxygen. So, in this way you can see that 1,3-biphosphoglycerate is converted into 2,3-biphosphoglycerate. इस तरह मुख्तलिफ बॉडी के जो पार्ट्स हैं उसके लिहाज से मॉडिफिकेशन आ जाती है, किसी भी पाथवे की आप बात करें. These are known as shunt reactions. So, this is a shunt reaction for the formation of 2,3-biphosphoglycerate is a shunt reaction for RBCs. Then in a very simple reaction, the phosphate group is moved from carbon number 3 to 2 to form the 2-phosphoglycerate. It helps in the reaction which will proceed from now onwards. Then dehydration of the 2-phosphoglycerate occurs, another very important reaction, because it gives rise to the formation of phosphoenol pyruvate, a very high energy compound, and this high energy will be used in the formation of ATP in the succeeding reactions. Now, you can see here, as I mentioned you, in this succeeding reaction, the phosphoenol pyruvate will be converted into pyruvate, the final product of the glycolysis. Here you can see again that ATP is converted into, ATP is formed here again, because this is an energy yielding reaction. So, 2 new ATP molecules are formed now. So, there is a gain of 2 ATP molecules in glycolysis. 2 molecules were used, 2 were formed, so this was balanced out, and then these are the 2 molecules which is a gain of the ATP molecules. The enzyme used here is pyruvate kinase, and here the fructose 1,6 biphosphate acts as the positive regulator, and this is an example of the feed forward mechanism, the feed forward way of regulating the enzymes. First, before it you have been learning about, or you have been listening the term feed backward. Now, it is feed forward, that product of a previous reaction can activate or regulate the reaction which is coming later on. So, this is an example of feed forward regulation. Fructose 1,6 biphosphate is doing this. So, let us have some certain questions. We should ask ourselves these questions. Why only 2 ATPs, and why glycolysis? I mean, if there is only 2 ATPs you have to get at the end of the day, why then we need glycolysis? Why there is a need for the shunt reactions? There is, I mean, why we need a shunt reaction here? Why there is no need of mitochondria in glycolysis? How a 6 molecule glucose ended up in 3 carbon molecule of 3 carbon molecule like pyruvate? What is its significance?